While a poppy stands as a solitary bloom, a sunflower head functions as a dense colony of hundreds of tiny individual florets. This complex arrangement on a specialized branch system is called an inflorescence. It relies on a structural hierarchy: the main axis, or peduncle, and the individual flower stalks, known as pedicels. The architecture of any inflorescence is determined by a single biological decision at the shoot apical meristem. The tip either keeps growing upward indefinitely, or it transforms into a terminal flower bud and stops. This growth decision separates the botanical world into two distinct categories: Racemose and Cymose. In a racemose inflorescence, the main axis never terminates in a flower. This allows for indefinite vertical growth as the peduncle extends. This creates acropetal succession. Since the tip is always moving upward, the oldest flowers remain at the base, while the youngest buds are found at the apex. This axis expansion ensures a staggered blooming period that moves sequentially from the bottom to the top. Racemose types are differentiated by the stalks of the individual flowers. A typical raceme has stalked flowers, while a spike consists of sessile flowers attached directly to the axis. In this illustration of Cassia fistula, the yellow flowers are held by long, prominent pedicels, identifying it as a typical raceme. Shape variations depend on how these stalks elongate. If the lower stalks grow longer to create a flat top surface, it is a corymb. If every stalk emerges from a single node like an umbrella, it is an umbel. In the capitulum seen in sunflowers, the main axis flattens entirely into a disc to hold its crowded florets. Despite these different geometric arrangements, each one maintains indefinite growth of the primary axis. The Cymose rule works differently. Here, the main axis terminates in a flower, which stops vertical growth and forces new branches to emerge from lateral buds below. This results in basipetal succession. The terminal flower is the oldest part of the system, and younger flowers develop downward toward the base. Look at the center panel of this diagram. Note how the main stem ends in a single flower, with two symmetrical branches arising from beneath it. This is a cymose arrangement because the growth is determinate. It is physically limited by that terminal flower. Beyond the standard rules, there are special inflorescences, highly modified clusters that frequently appear on board and NEET exams. The cyathium uses a cup-shaped structure to enclose its flowers, while the verticillister forms condensed clusters at the leaf axes that can be mistaken for simple whorls. The hypanthodium, seen in figs, features a fleshy, flask-like receptacle where the flowers are hidden entirely on the inside. These extreme structural modifications serve specialized reproductive strategies, often involving specific pollinators. We can now confirm that sunflowers and figs are not single flowers, but highly organized communities. To identify any inflorescence, check one specific point: the apex of the main axis. If the apex continues to grow with younger flowers at the top, it is racemose. If the apex is a terminal flower with younger ones below, it is cymose. Specialized structures like cups or flasks indicate the special types. Understanding the behavior of that central growth tip provides a reliable framework for navigating the complexities of plant morphology.